The nervous system consists mostly of nervous tissue, which is highly cellular. Less than 20% of the central nervous system is extracellular space, which means that the cells are densely packed and tightly intertwined. Nervous tissue is made up of just two principal types of cells, neuroglia and neurons. Neuroglia are supporting cells. They are smaller cells that surround and wrap the neurons. Neurons are the excitable nerve cells that transmit electrical signals. There are 6 types of neuroglia, 4 in the central nervous system, and 2 in the peripheral nervous system. Astrocytes, oligodendrocytes, microglia, and ependymal cells are only found in the central nervous system, while Schwann cells and satellite cells are found in the peripheral nervous system. Overall, the functions of neuroglia are to provide a supportive scaffolding for neurons, to segregate and insulate neurons, guide young neurons to the proper connections, and promote health and growth of the neurons. Astrocytes are the most abundant, versatile, and highly branched glial cells. They cling to neurons and their synaptic endings and cover capillaries. Functionally, they support and brace the neurons, anchor neurons to their nutrient supplies, guide migration of young neurons, maintain the appropriate chemical environment for generation of nerve impulses, take up excess neurotransmitters, help to form the blood-brain barrier, and help the formation of synapses in learning and memory. Oligodendrial cells are myelinating glia in the central nervous system, while Schwann cells in the peripheral nervous system. They provide layers of membrane that insulate the axon, which are called the myelin sheath. The sheath is periodically interrupted by nodes of Ranvier. Myelin sheath speeds the propagation of nerve impulses down the axon. Neurolemma encloses outer nucleated cytoplasmic layer of the Schwann cell and aids regeneration in axon injury. The white matter in the regions of the brain and spinal cord is a dense collection of myelinated fibers, while the gray matter is mostly soma and unmyelinated fibers. Ependymal cells provide lining off fluid-filled ventricles within the brain. It directs cell migration during brain development. Microglia phagocytes to remove debris left by dead or degenerating neurons and glia. Satellite cells are a type of glial cell that line the exterior surface of neurons in the peripheral nervous system. They also surround neuron cell bodies with ganglia. They provide structural support and regulate the exchanges of materials between neuronal cell bodies and interstitial fluid. A prototypical neuron consists of 3 main parts: the soma, axon, and dendrites. The soma is a spherical central part of neuron. The cytosol is enclosed by the neuronal membrane. The soma contains the nucleus and organelles. It has well-developed endoplasmic reticulum, Golgi apparatus, mitochondria, and ribosomes. However, it has no centrioles, hence its amitotic nature. There are 3 types of cytoskeleton that holds the neuronal membrane. Microtubules, which run longitudinally down the neurites, neurofilament, which are intermediate filaments in other cells, and microfilament, which are found throughout the neuron, particularly numerous in neurites. The axon starts from the axon hillock. No rough endoplasmic reticulum extends into the axon, and there are very few free ribosomes. The protein composition of the axon membrane is fundamentally different from that of the soma membrane. Axons may extend from less than 1 millimeter to 1 meter long. The branches of axons are called axon collaterals. The end of axon is called axon terminal. The site where axon comes into contact with other neurons or cells and passes the information to them is called a synapse. Branches at the end of axon that forms a synapsion, dendrites, and cell bodies in the same region are called terminal arbor. The cytoplasm content is different between axon and axon terminal. Microtubules do not extend into the terminal. The terminal contains numerous small bubbles of membrane called synaptic vesicles. The inside surface of the membrane that faces the synapse has a particularly dense covering of proteins, and it has numerous mitochondria. Dendrites of a single neuron is called a dendritic tree. The wide variety of shapes and sizes of dendrites is used to classify groups of neurons. Dendritic membrane under the synapse has many specialized protein molecules called receptors that detect the neurotransmitters in the synaptic cleft. Electrical signals in dendrites are conveyed as graded potentials and not action potentials. Neurons can be classified according to the number of neurites, such as unipolar, bipolar, or multipolar. The dendrites, such as stellate cells or pyramidal cells. The connections, such as primary sensory neurons, motor neurons, or interneurons. The axon length, such as projection neurons or circuit neurons, or neurotransmitters, such as cholinergic, adrenergic, or dopaminergic neurons. If you like this video, please hit the like button below, and subscribe to my channel to see more videos in future. Thank you for watching. Bye.